Have you ever wondered how we all look similar, even though we all experience different developmental environments and backgrounds? Animal development produces consistent characteristics across individuals in a population. These characteristics are often robust in the face of environmental and genetic changes. How are developing organisms so reproducible in spite of environmental and genetic changes, and what are the mechanisms behind this reproducibility, and what happens when they break down? In the paper we recently published in Genetics, we investigate how changes in environmental temperature can affect cell fate outcomes. To study this question, we use the model organism Caenorhabditis, elegans, a small soil-dwelling nematode with a fully characterized genome and cell lineage. C. elegans has a population of cells that during larval development behave like stem cells. These are known as seam cells, identified here using green fluorescent protein. Seam cells have a very stereotypical division pattern at the normal growth condition of 20 degrees, dividing asymmetrically to produce hypodermal and neuronal cells throughout development, as well as symmetrically to expand the population of seam cells. Asymmetric distribution and activation of the cell signaling pathway, known as the Wnt pathway, provides the necessary cues for asymmetric cell division, while the Runx homolog, Runt1, and its binding partner, Bro1, are necessary for symmetric cell division, overriding the Wnt asymmetry at an early developmental stage to expand the population of cells. In this study, we raised C. elegans at 20 and 25 degrees and compared post-developmental seam cell number, usually an invariant 16 seam cells for each side of the worm. We found animals growing at 25 degrees had an increase in seam cells at the end of development. This error was localized to a particular seam cell in the posterior of the animal. These results showed that although seam cells appear very similar along the seam axis, they displayed different sensitivities to phenotypic pressure, such as changes in temperature. Subsequent loss-of-function experiments showed the Hox gene, MAP5, expressed in the posterior region where the error occurs, and the beta-catenin homolog BAR1, expressed in the seam cells, were required for errors in cell fate choice. We also show that Wnt activity at L4 was not as asymmetrically distributed at 25 degrees, and surprisingly even at 20 degrees. Further, we observed the GATA transcription factor, EGL18, involved in Wnt activation and usually asymmetrically distributed between seam cell daughters, now displayed symmetric distribution in a number of animals. Therefore, we suggest impaired cell signaling via the Wnt pathway leads to ectopic Wnt activation in anterior V6 daughters at L4 and a retention of the seam cell fate. Following these results, we investigated seam cell number in C. elegans isolates from around the world. From these isolates, we found varying sensitivities to seam cell errors at 25 degrees, with the Hawaiian strain CB4856 able to suppress errors in seam cell duplication. Our results suggest that cryptic genetic variation may be able to buffer the response to temperature-mediated phenotypic errors. These results leave open a few exciting questions. Why are certain seam cells more sensitive to phenotype pressures? Which are the responsible genetic loci and mechanisms for this developmental buffering? And can we identify previously unknown regulators of the highly conserved Wnt pathway by comparing isolates? Following these questions could lead to a greater understanding of evolutionary adaptation and even cancer survivability. This work was carried out at Imperial College London and funded by the ERC and Leverhulme Trust.